Hi everyone, welcome to Studia Academy Past Paper Walkthrough. In this video, we will be discussing IGCSE Edexcel Biology May 2024, Paper 2B. Before answering question 1, we must read the passage below. Let's use the information in the passage and our own knowledge to answer the questions that follow. Let's go through question 1. Part A, Megalodon was an animal. What feature does Megalodon share with yeast? Both are eukaryotes, so they have nuclei in their cells, and both store carbohydrate as glycogen. Fungi don't have nervous coordination, so that's out. The correct answer is B, statements 1 and 3. Part B. Scientists think that the giant predatory toothed whales that replace the extinct megalodon consume the same sources of food. Explain. How did toothed whales cause megalodon's extinction? You can read lines 5 to 7. The answer is, there was competition between them for the same food source. The whales were better adapted faster, better at hunting, so they outcompeted the megalodon, eating more food. That left less food for the sharks, so their population couldn't be sustained, and they went extinct. Part C1. Volcanoes release sulfur dioxide into the atmosphere. Describe the biological consequence of sulfur dioxide release. You can see lines 10 to 12. The answer is acid rain, which lowers the pH of soil and water. That damages habitats, kills aquatic life, and harms plants, including deforestation and death of fish in lakes. Part C2. The release of large amounts of dust into the atmosphere reduce light intensity. Explain why this would cause a loss of food chains. You can look at lines 9 to 11, because the light is needed for photosynthesis in producers like plants and algae. Less light means less photosynthesis, so less glucose is made, and producers die. Since producers are the base of all food chains, no energy enters the food web, so the whole chain collapses. Part D1. Explain why the cells in cryozoos are put in salt and sugar solution rather than pure water. You can read lines 16 to 18 for the answer. In pure water, water would enter cells by osmosis because the cell contents are more concentrated, and the cells would swell and burst. The salt or sugar solution is isotonic, so there's no net water movement and cells stay intact. Part D2. Describe how scientists could clone a bunting. Look at lines 18 to 20. The cloning process is actually very simple. Step 1. Take a body cell, a somatic cell, from the banteng. Step 2. Take an egg cell from another species and remove its nucleus, enucleation. Step 3. Insert the banteng's nucleus into the empty egg. Step 4. Give a mild electric shock to stimulate the egg to divide, forming an embryo. Step 5. Implant the embryo into a surrogate mother to develop. Last part of question 1. Part E. Explain why releasing cloned mammoths could have negative and positive effects on Arctic biodiversity. You can see lines 22 to 28. On the positive side, the passage says mammoths helped keep soil frozen, which stopped methane from being released. Methane's a greenhouse gas, so that's good. They also added nutrients to the soil, which could help more plants grow more biodiversity. But on the negative side, the Arctic today is different. Their natural predators are gone. So mammoths might overpopulate, eat too much vegetation, and damage plant communities. They could also outcompete animals that currently live there, pushing them out. Let's move on to question 2, which is all about excretion and osmoregulation in plants. Part 1. You're asked how to set up the apparatus to test if light intensity affects water loss. Here's the method. First, cut the shoot underwater, ideally at an angle, to prevent air bubbles from getting into the xylem. Dry the leaves, check all seals are watertight, and use petroleum jelly if needed to make sure the bung is in properly. Next, introduce an air bubble into the capillary tube. That bubble is your marker. Then place a lamp at different distances from the shoot to change the light intensity. For each distance, measure how far the bubble moves in a set time. Say 5 minutes. Make sure you control other variables like temperature, humidity, and wind speed. Repeat each measurement and calculate a mean. You can use a reservoir to reset the bubble between runs. Part 2, the dependent variable here is simply what you're measuring. That's the volume of water lost, which you see as the distance the bubble moves, or the rate of bubble movement. Part B1. The table shows more water lost at higher light intensities. Why? Because light stimulates stomata to open for CO2 uptake during photosynthesis. When stomata open, water vapor diffuses out, so transpiration increases. As light gets brighter, stomata open wider, until they're fully open. Part B2. Now, a desert plant gives very different results. It loses much less water, and at high light, it loses almost none. That's because desert plants have special adaptations to conserve water. For example, they have sunken stomata, a thick, waxy cuticle, and fewer stomata overall. Many desert plants only open their stomata at night when it's cooler, so during the day, stomata stay closed, meaning no transpiration in high light. This stops the plant from wilting and going flaccid. Now, let's take a look at question 3. Part A asks us why athletes lose water when they exercise. When you move, your muscles generate heat. Your body recognizes the rising temperatures and starts sweating. As sweat evaporates off your skin, it takes heat with it. That's your body cooling you down. So water loss is actually part of temperature control, or homeostasis. To answer part B1, start by looking at the table. The athlete who had no drink produced only 100 cubic centimeters of really dark urine. What's the story? They're dehydrated, so their body goes into conservation mode. The brain detects low water levels and tells the pituitary gland to release more ADH, antidiuretic hormone. A DH is a hormone that signals to the kidneys to reabsorb more water, so instead of peeing it out, the body holds onto it. That means a small volume of urine, and because it's so concentrated, it's dark yellow. Part B2. Compare the isotonic drink athlete to the pure water athlete. The pure water one produced 750 cubic centimeters of very light urine. That's because pure water dilutes the blood quickly. The body recognizes that water levels get too high and gets rid of the excess, so color of urine is lighter. The isotonic drink, though, has the same concentration as blood, so it doesn't cause that rapid dilution. It gets absorbed nicely, replacing both water and the salts you lost sweating. Result, a moderate 500 cubic centimeters and yellow urine. Not too dark, not too light. Moving to question 4. Part A1. Where is LH made? It's the pituitary gland, that little gland at the base of your brain. Part A2 asks us what LH does in the menstrual cycle. LH has 2 main jobs. First, halfway through the cycle, a big surge of LH triggers ovulation. That's the release of an egg from the ovary. Second, after ovulation, LH tells the empty follicle to turn into the corpus luteum, which then churns out progesterone to maintain the uterine lining. Pretty important stuff. Part B. How do amniotic fluid and the placenta help the fetus grow safely? Amniotic fluid is like a personal bodyguard. It's a shock absorber. If mom bumps into something, the fluid cushions the baby. The placenta is the life support system. It's where diffusion happens. Oxygen and nutrients like glucose pass from mom's blood into the baby's blood, and waste like urea and CO2 go the other way to be removed. Part C1, a quick maths bit. We need the percentage difference at 36 weeks between Group A and Group D. From the graph, Group A is about 3,300 grams. Group D, about 2,600 grams. The difference is 700 grams. Formula, difference divided by original, times 100. So 700 divided by 2,600 times 100 equals about 27%. Part C2. What do the graph and your knowledge tell us about smoking and supplements? First, let's look at smoking. Compare Group A, non-smokers with supplements, to Group C, smokers with supplements. The smokers' babies are smaller. Why? Smoking narrows blood vessels, so less oxygen and nutrients reach the baby. Now look at supplements. Compare Group A, supplements, to Group B, no supplements, both non-smokers. Group B babies are smaller, so supplements clearly help growth. But here's the kicker. Compare Group C, smokers with supplements, to Group B, non-smokers without supplements. Group C babies are still smaller. That tells us smoking cancels out the benefit of supplements. Next, question 5, Part A1. We've got one DNA strand, TACCG-TAATTAT. What's the complementary strand? Just remember the pairing rules. A with T, C with G. So T pairs with A with T, C with G, and so on. That gives us ATGGCATTAATA, which is option A. Part A2. Which order is correct for protein synthesis? It's DNA to RNA to protein, and the processes are transcription, DNA to RNA, then translation, RNA to protein. So that's option A. Part B1. This is a bit like a puzzle. The sequence WXY from UUUAU GU. Check the table. W can be UUU or UUC. 2 options. X is only AUG. 1 option. Y can be UGU or UGC. 2 options. So total combinations equals 2 times 1 times 2 equals 4. They already used 1, so there are 3 other combinations that give the same amino acid sequence. Part B2. This is a key idea. Most mutations don't change anything. Why? First, the genetic code is degenerate. That's a weird word, but it just means most amino acids have multiple codons. So if a mutation changes UUU to UUC, it still codes for the same amino acid. No change to the protein. Part B3. How can mutations happen more often? Exposure to mutagens, things like radiation, UV light, X-rays, or certain chemicals, like in tobacco smoke. Let's finish with question 6. Part A. 1 gives us percentages. Lipid 16, protein 38, carbohydrate 16. Find the ratio 1 to n to 1. Divide everything by 16. 16 divided by 16 equals 1. 38 divided by 16 equals 2.375. 16 divided by 16 equals 1. So n equals 2.4 if we round it. Part A2. This question wants us to answer about the ethanol emulsion test. Rub the sample with ethanol to dissolve any fats. Then pour that ethanol into water. If lipids are present, you'll see a milky white emulsion, like cloudy water. Clear water means no lipids. Part A3. This question asks for one function of protein in the body. The answer could range from the growth and repair of tissues, or making enzymes, hormones, or antibodies. Moving to Part B1. The starting mass is 1,250 grams, end mass 3,750 grams. Gain equals 2,500 grams over 7 months. 2,500 divided by 7 equals 357 grams per month, to 3 significant figures. Part B2. This question is simple. All we need to do is rearrange. Mass of pellets equals index times mass gain. For non-GM, 1.9 times 2,830 minus 1,250 equals 1.9 times 1,580 equals 3,002 grams. Part B3. GM fish have a lower feed conversion index, 1.2 versus 1.9. Why? Basically, they're more efficient. They turn food into body mass better. Maybe they digest food more completely or have a faster metabolic rate. They waste less energy, so they need less food to grow the same amount. Part B4. Name one other abiotic factor they should keep constant. Could be temperature, oxygen levels, pH of the water, or light. Anything non-living that could affect the fish. Finally, Part B5. This is a big evaluate question, worth 5 marks. Scientists say a GM tilapia are better for the environment because they'd cause less nitrate and less disease. We need to weigh it up. Supporting the conclusion, look at the data. GM fish have a lower feed conversion index, so they need less food. Less food means less waste, fewer fish poops, and less leftover food. That means less nitrate going into the water, which reduces the risk of eutrophication, where algae grow like crazy and suck up all the oxygen. They also grow faster, so farming cycles are shorter, less impact over time. But here's the counterargument. The conclusion ignores other stuff. What if GM fish escape? They could breed with wild populations and mess up the gene pool, reducing genetic biodiversity. And the less disease claim? That's not backed by the data. It's just an assumption. In fact, if they're all genetically identical, a disease could wipe them out easily. Thank you for watching. Education made easy with Studia.